Good afternoon, everyone. Today, we'll explore the fascinating world of enteroviruses, focusing specifically on Echovirus 9, a member of the enterovirus B species within the Picornaviridae family. These small, non-enveloped positive sense RNA viruses are responsible for a range of diseases, including viral meningitis. Enteroviruses are highly adaptable and primarily transmitted through the fecal oral route. They can also spread via respiratory outlets or contact with contaminated surfaces. Structurally, these viruses are about 30 nanometers in diameter and have an icosahedral shape. Their protein shell, or caspid, made up of four proteins VP1, 2, 3, and 4 that encapsulates their RNA genome, providing stability and protection against environmental stresses. Echoviruses are a significant subset of the enterovirus B species. The term echovirus stands for enteric cytopathic human orphan virus, reflecting their initial discovery as an orphan viruses, unassociated with any known disease. Among the echoviruses, serotypes like echovirus 6, 9, 11, and 30 are well known, each identified by specific antigens on their surface that trigger distinct immune responses in the host. Unlike most organisms that follow the linear path of DNA to RNA to protein, Echovirus 9 and RNA virus alters this process slightly. Its positive sense RNA genome directly serves as mRNA once inside the host cell. This RNA is translated by the host's ribosomes into a single large polyprotein. Viral proteases then cleave this polyprotein into several functional proteins, including structural proteins for the viral capsid and non-structural proteins crucial for replication. The non-structural proteins work together to form a replication complex that synthesizes a complementary negative strand RNA. This negative strand then acts as a template for the creation of new positive strand RNA genomes. These new genomes are either packaged in new virions or translated to produce more viral protein, thus continuing the infection cycle. Echovirus 9 initiates its life cycle by attaching to specific receptors on the host cell surface. The primary receptor used by E9 is the decay accelerating factor, DAF, a glycoprotein anchored to the host cell surface via GPI. DAF is widely represented on epithelial cells in tissues like the skin and the lining of the gastrointestinal tract. The binding of E9 to DAF is critical as it determines the virus's ability to attach and to invade the host cell. After E9 binds to DAF, it triggers a series of signaling events within the host cell. The first of these is cytoskeletal reorganization, where small GTPases such as RAC1 and CDC42 are activated. These GTPases play a vital role in rearranging the host cell's actin cytoskeleton, promoting the formation of membrane protrusions and ruffles. This reorganization is crucial for moving E9 towards areas of the cell membrane where endocytosis can occur. E9 primarily enters the cell through clathrin-mediated endocytosis. In this process, clathrin proteins assemble into a lattice structure on the inner surface of the plasma membrane, forming a coated pit around the bound virus. Adapter proteins such as AP2 link clathrin to the viral receptor, ensuring the correct formation of the vesicle. The clathrin coated vesicle then folds in on itself and pinches off from the plasma membrane, forming an endocytic vesicle that encloses E9.
Now for uncoating. After entering the host cells via clathrin-mediated endocytosis, it is trapped inside an endosome, a sort of cellular compartment. As the endosome becomes more acidic, dropping to around a pH level of 6, the virus senses this change. In response, the E9 viral capsid, which is essentially the protein shell of the virus, undergoes a structural transformation. This change exposes hydrophobic or water-repelling protein domains on the capsid. These domains interact with the endosomal membrane, causing it to form small pores. So, through these pores, the E9 RNA is released into the cytoplasm, the fluid inside the cell. This release is done in a way that protects the RNA from degradation, ensuring it can begin its job of replication. Lastly, for echovirus 9, the icosahedral structure is fully maintained throughout, and an empty particle would remain after the E9 RNA have been externalized into the cytoplasm. Now, onto transcription. Normally, when a cell reads genetic instructions, it starts by copying a specific part of the DNA sequence, known as a gene, into an RNA sequence. For most DNA viruses, this is a crucial step because they need to transcribe their DNA into mRNA before any proteins can be made. However, echovirus 9 or E9 operates differently. Since E9 is a positive sense single-stranded RNA virus, it does not need to go through transcription at all. The positive sense RNA acts as both the genetic material and messenger RNA or mRNA. This means as soon as E9's RNA enters the host cell, it can immediately be translated into proteins by the host ribosomes, bypassing the transcription step entirely. Now, onto translation. Unlike most mRNAs that have a 5' cap and a 3' poly A tail, which are structures that help with the initiation of translation, E9's mRNA lacks these features. Instead, E9 relies on something called an internal ribosome entry site, or IRES for short, located in its 5' untranslated region. The IRES interacts with the host cell's translation initiation factors and ribosomal RNA, forming a scaffold that recruits the ribosome. This bypasses the need for a 5' cap and allows the ribosome to start translating the RNA directly into a polyprotein. This polyprotein would then be cleaved by viral proteases into individual functional proteins that are essential for the virus's replication. Now, onto genome replication. So, what makes E9's genome replication different from other RNA viruses? One key difference is that E9 carries the genetic information for a special enzyme called RNA-dependent RNA polymerase known as 3' pol. This enzyme is critical because it synthesizes both the positive and negative RNA strands necessary for replication. For this synthesis to occur, a primer is needed. In E9, a small viral protein called VPG or viral protein genome linked is modified to act as this primer. The RNA polymerase then uses a secondary RNA structure called the cis-acting replication element or CRE as a template. Now, with VPG serving as a primer, the RNA polymerase elongates the RNA to form a negative stranded intermediate. This negative stranded RNA cannot be translated directly into proteins but serves as a template for making more positive stranded RNA. These new positive stranded RNAs can then be used to produce viral proteins or be packaged into new virions, the complete viral particle that will go on to infect other cells.
For viral latency, the echovirus 9 is primarily known for causing acute infections, but recent research has revealed that it may exhibit a latency-like state under certain conditions. This means that the virus can remain dormant in specific tissues, such as the central nervous system, the myocardium, and the gastrointestinal tract for extended periods. During this latent phase, the virus avoids detection by the host's immune system, remaining quiescent until factors like immune suppression or cellular stress triggers its reactivation. This ability to persist in a latent-like state might be linked to the virus's ability to hide within cellular compartments like autophagosomes. By manipulating host cell processes such as autophagy, E9 may protect itself from degradation, allowing it to survive and potentially cause chronic conditions like myocarditis. For virion assembly and maturation, after E9 replicates its genome within a host cell, the next critical step is the assembly of new virions. This process begins with the self-assembly of viral capsid proteins VP1, VP2, VP3, and VP4 into a pentameric structure, which then combines with other pentamers to form an isocoegal capsid. Inside the capsid, the viral RNA is inserted, creating a structure known as a procapsid. The maturation of these procapsids is a tightly regulated process involving viral proteases which cleave precursor proteins into their mature forms. A key step in this process is the cleavage of the VP0 protein into VP2 and VP4, a transformation that is essential for stabilizing the viral capsid. Without this cleavage, the virion will be structurally unstable and non-infectious. Proper maturation not only stabilizes the virion, but also exposes regions of the capsid necessary for binding to host cell receptors, which is critical for the virus to successfully infect new cells. For the release of new infectious virus, once the virions have matured, they must be released from the host cell to continue the infection cycle. The most common method for E9 to release new virions is through cell lysis. As the number of virions inside the host cell increases, the cell's machinery becomes overwhelmed, leading to the disintegration of the cell membrane. This cytolytic release causes the host cell to burst, releasing the virions into the extracellular space where they can infect nearby cells. This rapid and destructive method of release is characteristic of the acute phase of E9 infection. However, recent studies have identified alternatives like non-lytic release mechanisms. One such method involves the packaging of virions into vesicles, such as autophagosomes, within the host cell's cytoplasm. These vesicles eventually fuse with the plasma membrane, releasing the virions without immediately killing the host cell. This non-lytic release is less inflammatory, allowing the virus to evade immune detection and potentially establish persistent infections. Additionally, E9 is shed in large amounts in stool, making the gastrointestinal tract a significant site of replication and transmission, especially through the fecal-oral route. This shedding can occur even during asymptomatic infections, contributing to the virus's persistence in the environment and its potential to cause outbreaks. Next, we have mutation. As an RNA virus, Echovirus 9 has a high mutation rate, driven by its RNA-dependent RNA polymerase, an enzyme that lacks proofreading ability. This high mutation rate results in a diverse population of virions, known as quasi-species, within a single host. This genetic diversity provides E9 with a rapid and effective means of adapting to selective pressures, such as the host immune response or antiviral treatments, increasing its chances of survival and propagation. Some of these mutations can lead to antigenic variations, where changes in the viral capsid proteins allows the virus to evade neutralizing antibodies. This process of antigenic drift is a common strategy among RNA viruses to avoid the host's immune system, leading to the emergence of new virus strains capable of causing outbreaks. While many mutations are neutral or have no significant impact on the virus's biological activity, some can enhance its virulence or alter its ability to infect specific cell types. For instance, mutation that changes the viral receptor binding sites may increase the virus's ability to infect cells or avoid neutralization by antibodies. In addition to point mutation, E9 can also undergo recombination with other enteroviruses. This process occurs when two different viral genomes co-infect the same host cell, leading to the exchange of genetic material and the creation of new viral strains with varied pathogenicity, host range, and antigenicity. Recombination plays a specific role in the evolution of enteroviruses, contributing to their genetic diversity and adaptability.